So I'm Rosana, Rocha. I'm from Brazil. I work at the Federal University of Panama, this is in South Brazil. And I work in Tunicate with taxonomy and bioinvasion. So here we had this workshop with 6 instructors. It was me and one from Spain, one from Japan, and the others from the States. And we had 18 students from all the Americas and also from South Africa and Singapore. And we're doing a 3-week course. The first week was just for taxonomy, so they know the animals of the region. And the second week we had more general biological studies that use tunicates as models, like ecology, genetics, and immunology, and molecular data comparisons and things like those, phylogeny too. And then the third week the students had the opportunity to do their own research, so they would choose theme subjects they liked most during the classes, and they did their own projects. They showed us all their results this morning, and there was a lot of collaboration between the students, and what we hope with this Tunicate course that they will still collaborate in the future between them, the students themselves, and within us professors, instructors too.
Each one is an actual organism, and they're hermaphrodites, so each organism has eggs and sperm, and they're sessile, which means they don't move as adults, and they just sit attached to whatever they're attached to and filter feed, and they'll react, like they'll close their siphons if you touch them, or
come over here.
Various things happen, and then they can release their eggs in these cases, and their sperm through one of their siphons into the water column, and then they meet in the water column and fertilize. And then there's other ones, like on the outside of this is just one tunicate, but there's also this slimy, colorful stuff is another type, and that's a colonial tunicate. So in that case, there's one tunic, but there's a bunch of little sort of organisms within each tunic, but they're all clones of each other, so it's hard to say whether it's one or multiple animals. And then they usually don't release their eggs and sperm. They brood their larvae somewhere inside their colony.
Where is the tunic itself?
So the tunic on these ones, the outer coat is the tunic. So if you cut away the tunic, then inside you have sort of a softer, more jelly-like organism with all the organs inside. So the tunic's protected, it's made of cellulose. And then in these ones, basically most of this, what you're seeing is tunic, and then these little tiny dots you see are the zoids, which they look like little miniature ones of those.
Essentially what we're doing is we don't have actual substrate in the dishes. What we're really doing is we just have chemical essence of those substrates. So basically the whole idea is that they're settling in the bottom of a petri dish, but they don't, versus the control, the only difference is the actual chemicals in the water. So So I guess so far we've seen, you know, about 50%, 40% settlement in the dishes just with seawater, and in the dishes with the experimental treatment, all three different types are seeing like 80 to 90% settlement. So.
And what defines settlement?
So settlement is, there's a little swimming larva, and they kind of swim around pretty spastic and move, like basically what they'll do is swim toward the bottom, and they'll use these adhesive glands on the front of their head to kind of stick to the substrate, stick to the bottom, and once they stick to the bottom, they'll retract their tail, so they'll basically resorb their tail and start going through metamorphosis, which basically causes they form their siphons that they'll use for filter feeding as an adult, and they'll kind of move those up to the the top of their head that's already cemented in, or I guess it's moved to the old posterior and make kind of a new entire body.